Yeah, we still have a few space in the back, in the front here. And I promise Vin de Villas, he did have a shower yesterday, so we can have some space here. Yeah. Yeah. No, so joke apart, yeah. We, we we are really excited about the seminar today. Not only am excited about the presenter, which you heard a lot in the next half an hour, 40 minutes, but also about our own people. So this seminar, it is what, as everyone know, we run many external seminars every month. Yeah, we are receiving some of the top scientists here, but that's start a new wave when our own young scientists, yeah, is starting organizing and invite guests. So this seminar, which the great president is going to see, I have nothing to do, but that's Dr. Marais Hofstra, yeah, which is a medical microbiologist and also a medical doctor in our group, that have set up a very impressive collaboration with Ruan Marais that invited him to give a presentation to us. So Marais, if you can introduce your guest, and we're also looking forward for our other scientists to organize such seminars also. Thank you.
Yes, very exciting that you're here. So just to introduce to you all our speaker of today. He is a medical doctor. He did first a specialization in virology, but then at the end he also did a track in microbiology and realized that he preferred that, so he decided to also do a specialization in microbiology. He has done an MSc in virology in Imperial College in the UK, and is doing a PhD in bioinformatics. So it's basically combining all the different skill sets that you need to be starting working on this metagenomics to really bring that towards a clinical diagnostical tool. So it's very exciting. We have been mainly working with VirCap-seq. Dr. Marais is really working on BACCAP-seq, so very much focused on bacteria, and we're very excited that we're hopefully going to start collaborating and getting more experience together in that. And we're looking forward to your talk today.
Thank you. Thanks so much for the invite, and I hope I can give you some interesting things to think about, and about where clinical diagnostics and sequencing can merge to really give good outcomes. So I'm going to be talking about building a better lab. So working my way through my reg time in virology and then in microbiology, really I saw how, first starting in virology, how everything had moved from culture-based and cell culture, and had transitioned first to everything is a real-time PCR, and is gradually transitioning to where sequencing is really coming to the fore. And then went into microbiology, where everything is grown on a plate, or almost everything. So I started to really wonder, is it time to replace the phenotypic microbiology lab? Can we take a whole collection of samples, run it through an automated platform, and really get the additional information out? So we started really simple with just some shotgun metagenomics on central nervous system samples, which is really quite a low-hanging fruit, and really found good results. Even with very low depth, you get very good sensitivity and specificity. And this study we did in patients that had lumbar punctures in Groote Schuur Hospital, and we followed up the routine testing, and we did relatively low depth shotgun sequencing, but you can do that with CSF, and found it really performs really well. The only things that we didn't detect were things that were largely diagnosed with serology, like syphilis, and that was a major limitation. We did another study more in microbiology, where we did the CERI molecular epidemiology, and I mean this figure is not for you to unpack individually. It's really just to show you this is the amount of information that you can pull out of a bacterial isolate if you sequence it versus just growing it on a plate. Sorry?
What kind of information is that?
So this is whole genome sequencing.
Yeah.
All right. So at the first row, basically, this is the hospital. that the sample is taken. So you can see Tygerberg, Red Cross Hospital, Groote Hospital, the sequence type, the K locus, the O locus, what resistance genes that were present in CRE. So these a lot had NDMs and OXA48. Combine that with the MIC information. These are a curated list of virulence factors. So, siderophores, which are important in Klebsiella, fimbriae, so adhesion factors, and then these genes here right at the end were to see whether we had any hypervirulent Klebsiella, which is really important, and we were extremely happy to find that so far, no, we did not have any, which was a good finding. But what you should take from this is, we can culture an isolate on a plate and we can say what it's susceptible to, but this is all the information that is still there that we could potentially use. That might be the difference in why we are treating someone with this drug, but it works 70% of the time. But why not the other 30% of the time? Potentially that information is available, but we just can't use it yet. So the next really important thing comes out of what other literature and what else has been done in microbiology over the last few years. And we're really starting to find that we are seeing ISO-certified genomics workflows that can predict with high accuracy whether an isolate is susceptible to a specific drug. So for example, here for Salmonella against agar dilution, which we don't even routinely do in our routine labs, you had 98.9% accuracy. And that's coming through for multiple organisms. Here for Neisseria gonorrhea, the genomics, whole genome sequencing based tools are really getting great accuracy with very low, very major error rates that you would expect, that you find is better than phenotypic, as well as for other organisms like Strep pneumoniae. You're really starting to get a critical mass of genomics-based tools that you can use to predict AMR. And if you compare that to what we currently use, like the Vitek and the BD Phoenix and e-tests, you're starting to find that the genomics workflows in many cases are exceeding the accuracy of these tools. So really, you have to consider, is this something that we should be moving towards? And I certainly think so. And then you also have the really important aspect where, okay, we can prove equivalence with genomics-based tools, but can we exceed what is possible with genomics-based tools over just culture-based diagnostics? And here you can do things like real-time sequencing, where if we have to culture an organism and then do susceptibility testing, that is 48 hours minimum. But if we can sequence from a primary isolate and start having results within the first 6 hours, then, well, that opens a whole nother field of what we can potentially do to improve outcomes. Now, what is often put forward as a reason to do phenotypic versus genomics is complexity. It seems really simple to say, oh, okay, we're going to sample something, we're going to grow it, and we're going to say treat with kefapine. That is difficult to convince someone that we need to replace something so simple. But it is so much hidden complexity that people don't realize. Remember, that culture-based step, that is with standardized quality-controlled media, near universal procedures for sample collection, processing, transport, inoculation, all produced by bodies like the CLSI or UCAST, who give criteria for interpretation, which is produced in a standardized report format with standardized meaning for what we report as sensitive and intermediate and resistant. And all of that is built within a system that includes clinician training, that includes clinical trials that all feed into that system. So really, it's a misnomer to think that, oh, okay, well, we can't really replace this system because, how can we replace something so complex? Well, how can we replace something so simple and with something so complex? But really, the issue is that complexity of genomics can in the same way be standardized and normalized as it has been done with phenotypic microbiology over the last 200 years. So that is what I have been trying to do, or that is my aim in my research, is what needs to be standardized, what needs to be optimized, where do we need to reduce the costs, where do we need to improve or simplify analyses. So that's what I'm going to talk about for the rest of the talk. So really the first major barrier with clinical metagenomics, and here I'm talking about the goal is we need to do it directly from sample. That if we're going to get the benefit, we need to do it directly from sample. So we start there. You know, we can do whole genome sequencing directly from isolates, and we can immediately get benefits from that. But how do we get it to the point that we can do it from sample where we get optimal benefit? And really the biggest wet lab challenge there is sensitivity. So when you sequence a primary sample, the vast majority of the sample is going to be human DNA. So we need to address that. So there are ways to do that. You can deplete it, the human DNA, so that the organisms and the AMR genes are in greater abundance. We can enrich the organisms and the AMR genes and keep the host the same, or we could do both, right? We can deplete and we can enrich to get optimal information out of the sample. Now, I have gone with that approach and say, how do we best deplete and enrich, and how do we include that in the workflow? So we started with CRISPR depletion. And the reason we picked CRISPR depletion is because while there are various other methods like DNase treatment of samples directly in samples, saponin treatment, all of these will lose certain things. So let's say herpes viruses. These are important, yes. They're especially from blood, CSF, they're all important specimens. But what you have to realize there is that in clinical samples, the majority of DNA associated with those herpes viruses, which is DNA virus, is free DNA, short fragments of free DNA, not in a virus. So if you use a DNase treatment step for that, or a saponin treatment step, gone. So for those reasons, and to maintain the unbiased nature, but yet have good depletion, we went with CRISPR, and this is in collaboration with Philippe Selhorst at the Institute of Tropical Medicine in Antwerp. So he developed a protocol for using CRISPR that is based on the DASH protocol, which essentially is that you use CRISPR to deplete your library before you sequence it. And that's very, very basically. So we kind of, in that same line, decided, well, we would like to do this, but how do we best design those guides? So I wrote an automated pipeline to do that. So realizing that, you know, if you want to use this, how do you optimally decide which guides do you use? Now, you can do that by reading the literature and saying, well, these are the important genes. But we took a step back and said, well, let's rather do this in a completely unbiased way, so that you can take whatever sample media host background that you want, and we can design the optimal set of guides for that sample. Now, the reason that that's useful is that in human metagenomics, yes, you can design quite a good panel based on what is known about what genes are commonly found in an RNA-seq experiments, for example. But what if you want to use a bat sample? Or what if you want to use a tapir sample? Or what if you want to use a llama sample? Like, we don't know what, you know, there might just not be good information about what you should deplete. So what we did was we wrote some software that basically you put in whatever sample you want, right? And you give it the whatever database you don't want to deplete. So here you would, for example, use the RVDB database, right? And it will design the guides for you, exclude any offsite cuts, and provide you basically an order list of this is the sequence you just need to submit to IDT, and you can design your guides. And it will also tell you exactly which viruses there might be offsite cuts for your CRISPR panel in. And then we'll also give you output on how much depletion theoretically you will get in your sample with various conditions that you can set in the software. So for example, here is an experiment that we did on mouse sequencing experiment, basically, or mastoma, so technically not a mouse. And it will tell you where those cuts are going to be in the relevant genes. And you can also in silico predict the amount of depletion that you'll get from the library. So you can decide, you know, okay, we want to use 150 guides. That's going to give us 55% depletion of whatever we don't want. And the reason that this is a useful approach is because this is extremely cheap, right? Once you can produce these guides with through transcription-mediated amplification, you know, you can get sample costs way down below what you would need to do if you were going to do, for example, probe capture. Now, the other approach, however, is enrichment. Now, for certain things, you can't necessarily just do depletion. Now, if you are specifically think what you're looking for is RNA, then depletion is great, because in a RNA-seq type experiment, mostly you're going to be looking at ribosomal genes and mitochondrial genes. But if you're looking at a tissue sample and you want AMR genes, there aren't specific targets that can just be depleted and that will give you a lot of added sensitivity. So for those specific cases, you do need to do some form of enrichment. So I've collaborated with Columbia University's Global Alliance for Preventing Pandemics and looked at BACCAP-seq and VRCAP-seq, which essentially are probe sets that cover, for VRCAP-seq, all vertebrate viruses, and for BACCAP-seq, the core genomes and AMR genes for relevant, medically relevant and other bacteria. And we applied this to some orthopedic samples. So here you can see, despite the enrichment, you do still get a lot of human reads and some unclassified reads here that was just not bacterial. But in this specimen, the culture detected an Acinetobacter baumannii, and very clearly here, after enrichment, we got that same Acinetobacter baumannii popping out at 9%. Now, 9% is not little in a metagenomics shotgun experiment. Well, in a shotgun sequencing experiment from a tissue sample, that Acinetobacter baumannii might have been at 0.001%. And here we've got it up to 9%, which is very much interpretable. And I'll show you the AMR genes just a bit later. So this was a clear sample that only a single organism was isolated, and that very clearly popped up as the dominant organism, with essentially in this experiment where we use bracken, basically 90% of the bacterial reads were Acinetobacter baumannii. So that really, I mean, it gives you quite good confidence that that is in the sample. And then here we looked at a different specimen where the specimen itself cultured a range of different pathogens. Because here the question was, okay, so are we just going to get one organism pop out, the dominant organism, and everything else kind of just at baseline? But no, that's not what we found. So this isolate actually cultured an E. coli, a Proteus, a Morganella, and anaerobes. And all of these came out reasonably well. The Klebsiella pneumoniae, that's probably conserved reads in the Enterobacterales. So I wouldn't necessarily say that was there, but essentially you still got good resolution for all of the different organisms that were cultured, if you compare that to the actual culture detection. And if you compare that to what the routine diagnostic testing that was done, sensitivity was great. Specificity was a bit down, but that largely came from us detecting spores, which we would have, well, you can clearly see these are things that would not have grown, and that's where that drop in specificity came out. But sensitivity was reasonably impressive. Now, those are the two techniques that have found large application or reasonable application in the field, right? But you know, while we can get low cost for the CRISPR approach, the probe capture approach is expensive. And now if you need to do that for every single specimen, that at this stage, it is still too costly for routine implementation for every specimen. So, so it costs about $200 per sample, approximately. But that does depend on, you know, your lab setup and what depth you need and so on. But that's a reasonable estimate, which is not bad. But I mean, a routine phenotypic test is probably going to cost you in a region of 100 Rand, maybe 100 to 200 Rand, depending on the test. So that's about 17 times cheaper at this stage. So that kind of led us to say, well, you know, okay, these technologies are feasible from the technical perspective of getting the information we need out, but it is still too costly. So we can wait for sequencing costs to decrease, but is there potentially another route that we can take? So there's a recent paper that was published using magnetic nanoparticles with specific peptides to enrich or accelerate the rate at which you can get phenotypic culture results from clinical specimens. And so took a little bit of the idea of that technique and applied it to clinical metagenomics, and asking the question of, can we use this same approach but for clinical metagenomics? And so what I've done is take a collection of peptides and antibodies, which can be used upstream of both culture and sequencing diagnostics, with a goal of essentially developing a PAMP capture panel. So the current enrichment techniques focuses on pulling out the DNA you want after your library prep. Now that is already after you've done a ton of amplification of a whole bunch of human DNA. And you also need to, if you want a specific bacterial species, you need to include probes for that species. While if you potentially go with a PAMP capture approach, so a pathogen-associated molecular pattern, and capture those bacteria or viruses or fungi directly from the specimen before you do the extraction, potentially you can get far better cost benefit rate, far lower cost for potentially the same kind of enrichment. And that is what I've been evaluating over the last few months. And you can see here, we get reasonable pull down with our PAMP capture panel for Pseudomonas aeruginosa. If you look at control, where that is basically saying that if there's no capture step, versus do we capture this from the specimen, and we reasonably pull down all of the bacteria, and it performs as well for some of the conditions for Staphylococcus aureus. And if you know about bacterial species, I mean, Pseudomonas, that is a gram-negative, the surface is entirely different from Staph aureus. And this is with the same peptide, you can pull down both and get enrichment from both, from the specimen, at a cost that is a fraction of what you would if you were to design a panel for every single gram-negative organism, which would be hundreds of thousands of probes. Here you can use a single peptide to pull down all bacteria with a specific surface structure. And that is what I've been evaluating in terms of the enrichment approach. So moving past that, let's say now you have isolated and optimally sequenced your organism and you have your data. You're still not out of the woods. There is a lot of data generated, right? So for example, this from that Acinetobacter baumannii isolate that I showed you previously, these are all the resistance-associated genes that we detected for that one isolate. Now, I mean, a clinical microbiologist might look at that and say, well, I have kind of an idea of what I can use, but that's going to take you maybe an afternoon of looking through literature and looking at specific publications about the penetrance of specific genes and so on. I mean, it's going to take you an afternoon. And then when we're signing out results in the clinical microlab, you're signing out like a result a minute, probably. You don't have the time to review a massive page of AMR genes. So what I've done is I have created a tool that does that for you, that applies optimal clinical microbiology expertise and essentially takes the sequencing data and converts that into a standard phenotypic microstyle report. So I'm just showing you the API backend here to give you an idea of the functionality, and then I will show you the front end moving after the presentation. So that I've written the backend for this tool, and at the moment I'm busy deploying it and writing the front end like software for it so that it can be used in our clinical studies that we plan to do with it in the next few months. So basically what this does is you essentially, as you can for any laboratory interface system, you can create your patients with specific criteria and details of your patients, whether you can give them only IV therapy or whether they can take oral therapy, etc., what their renal function status is, what their hepatic function, etc., right? So that the software can have a good picture in terms of making its inference, right? It pulls in all the results. It has tools to pull in results that can be created directly on the tool or through the RedCap API. And obviously, eventually, it would just interface with any EPR, electronic health record, depending on the system that it's implemented in, to pull in all the results. Like, for example, here you can see it's pulled in a blood culture result from Staph aureus. And the important thing is here, that it pulls in microbiology, like culture results, as well as sequencing-based results. Right? Then the important thing here is the interpretation, right? So it then gives you the option to specify the criteria, the setting, or the details that you want included in your interpretation. So, for example, do you want it to include unavailable drugs? So that might be, okay, let's say in your hospital you don't have Ceftaz avibactam available, but you still wanted to include it as a possible option should nothing else be available, so that you can know I need to source this from somewhere, or just use what is available in the hospital. Should it apply empiric settings? So, for example, this patient has just came in. Should I take into account what the hospital's epidemiology is in terms of recommending therapy? If there isn't enough coverage of a specific gene, for example, let's say it detects a salmonella, but there's not adequate coverage of the gyre gene, but you know in your hospital your ciprofloxacin rates are extremely high. So then it'll say, okay, well, we're applying empiric settings and we're not including that as an option. And then other factors like what level of risk do you want to take when looking at things like pregnancy and site suitability. So let's say, for example, it only is supposed to tell you if the drug is 100% active at a site. Like, let's say you have a central nervous system infection, and you are query wanting to use cefazolin, which the data is still not 100% clear whether that's acceptable to use for central nervous system infections. So all of these factors you can apply in terms of guiding it and how to make the interpretation about what drug to recommend based on all of the available microbiology and sequencing results for this patient. And then it does it for you. So for this patient, for example, there was an MRSA bacteremia combined with E. coli and Pseudomonas aeruginosa bacteremia, as well as E. coli bacteremia, with complicated antibiograms. And the tool will tell you should use vancomycin and meropenem for this. And you can see the dose, unit, and interval there. We'll pull in that information as well. We've just set up a collaboration with the pharmacology department at UCT to use the SAMP to populate that information as well. Now, I've looked at the antibiograms of these organisms, and meropenem and vancomycin in this case is appropriate. But really, the important thing is the explainability. So it will also tell you, I gave this recommendation for Staph aureus in blood based on this reference, right? The durations based on this reference, right, for the E. coli in blood. Explainability here, because this I included because the vast majority of calls that you'll get as a clinical microbiologist are, okay, I see the antibiogram says I can use this drug, but it doesn't say anything about that drug. Can I use that drug? That's not on the antibiogram. And, often you'll just want you want all of that information to be available, right? So that you don't have to provide all of that input. And that's why I included this. So, for example, whoever is using the tool can go look and say, well, I kind of want to use ampicillin, but it doesn't say anything about ampicillin. So can you use ampicillin? So it basically goes through all of the details for every single drug and explains why or why not you cannot use that. Obviously, this is from the API backend, and the front end displays it in a much more user-friendly way. But basically, it can tell you that, if we're looking just at the central nervous system Staph aureus that was isolated, can you use ampicillin? Well, from the perspective of whether the drug is available, yes. Allergy, yes. Pregnancy, yes. Hepatic, yes. Renal, age, site, yes. Ampicillin does cross the blood-brain barrier. Stewardship, no, because technically it is not the narrowest agent that was suitable for that. But more importantly, testing inferred or intrinsic suitability, ampicillin is not something you use for Staph aureus in this case, and that's why ampicillin was not recommended in this case. So you can, for example, go through your entire list and say, well, I kind of want to use this, or I'm currently using this, but the report says I should use that. Can I use XYZ? And you can go into the tool and will in detail explain to you exactly why you can't use that drug, why you can use that drug, so that it is imminently explainable. Now, you might say, well, this is probably like some large language model that I trained on huge amounts of data. No, it is entirely algorithmic. So one of the first issues I ran into with writing this algorithm is that intrinsically it's a traveling salesman problem. As the more isolates you add, now it needs to evaluate every single possible combination. And essentially that, if you know the term, the notation there, it's a computational complexity of n factorial, which means that is the worst one that you could possibly want, right? But through applying so here you can see as I increase the number of different isolates that it needs to consider. So it needed to consider a Staph aureus that was isolated 3 weeks ago. It needed to consider a E. coli in urine, a crab that was isolated, 3 months ago, a coagulase negative staph that came from a blood culture like 4 days ago. As you increase that complexity of analyzing this treatment recommendation, at around about 8 isolates that it timed our threshold, and it would take more than 100 seconds to make that call. But by applying various heuristics and improved data structures, I have it now that it essentially maintains that computation below or at around about 0.1 seconds to make an inference about what the optimal therapy is for all of that information about a patient. And that is basically what I've been working on, trying to get the costs down in the wet lab side by both using a depletion approach as well as a direct from sample enrichment approach, trying to get around using probes, which are quite expensive. And then from the analysis side, how do you reduce the complexity of these results? And through that, basically writing algorithms that allow those results to be filtered through and be reported as you would in a phenotypic culture result. So the next phase of the study is prospective recruitment, and to see if we prospectively collect these samples, apply these sequencing technologies, and essentially recommend the theoretical optimal drug to use in these cases. How does that compare to what we are, how does that compare to routine care? And then if that is favorable in one or all of the sample types, then potentially we will do a prospective study where we do the sequencing analysis and access to the tools in real time to see how it performs. Right, just acknowledgements. So this work is through a Crick Africa Network Fellowship, where I'm hosted at UCT. My mentors, Adrian Brink and Robert Wilkinson. And the algorithmic work, that is through a PhD that I'm doing with Darren Martin. Obviously, my collaborators at Global Alliance for Preventing Pandemics, so specifically Prof. Ian Lipkin, Ken, and Sam, that has the most interesting stories, right? And then Philippe, who developed the CRISPR wet lab protocols that I worked with to write the algorithms to automate that process. And then obviously various students, Marais, Mia, Luano, Pietru, and the Crick STPs, the genomics STP, chemical biology STP, and basically all my other clinical collaborators who are basically involved in collecting samples or planning studies, etc. So just as a last thing, we have 5 minutes left. So the front end, I am basically working on adding now the front end, the user interface to this API. So this is what it looks like at the moment. So the important thing is it's an integrated system that includes the sequencing component as well as the laboratory interface. So the permission structure flows through entirely. It's also entirely deployable on any system. So I can run it on my laptop, can run it on a cluster where it'll use Singularity, or it can run it on a web server, and you basically just, Docker, it's run using a Docker Compose file. So you can basically just deploy it very easily. Now, like for example, if we now wanted to upload a file, so we have to select the facility that file is associated with, and we want to upload a sample, right? Just Yeah, I'm very scared, very scared. Right, and then you can see this now uploaded as a sample, and we need to allocate it to something. So, for example, it will see that this is a read one, read two. So we want to allocate both, and we have to find a patient to allocate it to. So I've just put myself in here. Okay, and you can create now create a new sample, right? To say, okay, well, let's say this was a blood sample, right? All the read paths and things were already there at the correct place, right? And you can say which test date it was, and you have to allocate it to a sample sheet, which I created before. And there you go. You've created your sample, ready to run through one of the pipelines. So as I said, the tool is still being linked to the API, but what I can show you is, for example, all right, so that's the kind of like the clinical interface side where a clinician or lab tech would now upload a sample and run it through a pipeline. But it's also for use by the lab staff if you want to now write a new pipeline quickly, right, within this kind of containerized structure. So you can, for example, go to the analysis tools section, right, and want to create a new tool. So, okay, we want to take the raw data, for example, here. The tool is associated with test hospital, right? Let's call it something, you know, okay, can like specify, for example, you know, what the minimum number of bytes that need to come out there and any specific arguments that need to be associated with it. So this would be more used by like the somewhat bioinformatically competent staff, but essentially, you know, you don't need to be able to code to use this. So if you want to associate any files with that process, no, I don't want to, any specific references. So ones I've uploaded, for example, is a human reference. So say we want to associate the human reference with that like tool, any scripts that it needs to be able to run, right? No. Okay, the input data. So, okay, let's say, well, this is going to be Illumina. So it's going to be, for this raw data, there's going to be 2 files, right? Okay, and the file type is going to be FASTQ, right? And you know, we can subset. Okay. Okay, right, so it's fine. Okay. Then now we need to use a specific environment. So this is linked to biocontainers, so you can go find the environment you want. But ones that I've just added before is, okay, well, we want to use, you know, FastP or that one. So link that container. And then here you can now construct your basically command line tool. So, you know, FastP. Right. So, and I mean, if you wanted to change the order here, you know, you could, as you want. Right. And then basically you just finish, you upload that tool. Right now, that tool is available here as a specific containerized tool. And basically what that will do is when you now construct a pipeline, you just say, I want to run these 5 tools. It will then create those containerized environments, delegate, run it on whatever system that you're running this, basically multi-threaded or not, as you choose. It will infer the available resources that the system has and keep this all containerized so that you could run it on a clinical server, that you don't need to run it on, you know, don't need to go use CZID to run it, or, you know, Galaxy, where you don't necessarily have control over the processes. So I guess you can think of it in that line. It's basically a Galaxy that is already fully integrated into a laboratory interface that can report everything and interpret the results that are pushed back to the patient. But as I say, this is the part of the front end that I've added so far. So hopefully at the next meeting I can show you a full demo from sample right until the end and analysis. Because yeah, writing a front end takes a bit longer than I expected. Let me put it that way. But yeah, that's my talk. Any questions?
Can I start off the questions maybe?
Yes.
So as a microbiologist, one of the things, at least in the Netherlands, we do is also use your reporting of your susceptibility as a way of antibiotic stewardship. So we're actually not reporting everything we've tested because you kind of want to guide clinicians to use maybe that drug that is smaller spectrum than the broad spectrum. So your tool being able to show everything, data and everything, are you thinking of using this actually also as an antibiotic stewardship tool and guiding clinicians in that way?
It is possible as well. So it will report to you the best possible option, which includes antimicrobial, like the algorithm includes antibiotics, like antimicrobial stewardship. That is one of the parameters that is built in as an exclusion factor. So if ampicillin is suitable, it's not going to show you ceftriaxone as an option.
Yeah.
So that is already built in. But yes, you can potentially use this and integrate the system into like a pharmacy, for example, where, because I mean, in the background, every single possible combination and single or combination of drugs has a very specific score. So you can rank every possible option, kind of, is this option better than that option from every possible angle. So you could like have this running in the background of a pharmacy that can pull in from the EPR, like all available information about that drug. And before they dispense it, you could say, well, you know, there is a better option. Maybe you should just contact the clinician and tell them now in the last hour from the automatically linked sequencing infrastructure, a better option has become available. You should probably change your prescription to XYZ. And that can all run in the background automated because it's deterministic. It's not, there's no human interaction or classification needed.
Yeah. Right.
Hi, thanks for the wonderful talk. I'm curious, so the tool for identifying the resistance genes, which reference database does it use? Because for some of these, depending on which database you choose, you could get a whole different range of resistance genes.
Yeah, so for the moment, I'm using CARD, but that decision, sorry. People don't hear on. Sorry, yeah. So at the moment, I'm using CARD. But that decision was largely based on the fact that the backcap-seq, uses CARD. So, the data that is coming through and being used for the analysis is designed for CARD. So it's unlikely that there's other resistance genes that are not in there that are going to be coming out. So, I mean, that's the justification.
Just a quick one. I'm also curious about if you can just expound more on the prediction of phenotypic resistance.
Yeah.
It's a big issue I've seen in the lab where you have two isolates from pretty much the same sample, same sequence type, but you run Vitek on them and one is susceptible to something, one is resistant to something. So those discrepancies, how do you work around them in a situation like this?
Well, that is a issue, I guess, of what data you are going to use for your prediction. If you only have Vitek data, that is obviously problematic because you can't really make the call one or the other. I mean, I would say that is reasonably rare that is the case, but what I have focused on is making it multi-component. So it will pull in culture as well as directed testing as well as the sequencing results and consider all of those when basically making a call. So in that case, one would hope that, you tested the same isolate twice, the Vitek on the one is this and the other one is that, but the sequencing is more in favor of the one that you would hope that situation arises. If it cannot make a call, obviously, well, then there is the human element of it saying, well, it cannot make a reasonable best guess in this situation because of XYZ. But it is extremely transparent about how every decision is made. So you can go in and see, okay, well, actually, this one wasn't selected because of this specific reason, right? And I think then that allows you to make that call in that situation where your input data is just not usable. Yes.
Thanks very much for an excellent talk. Perhaps you could answer this question. You talked in the beginning about, time to result is so important. But we know with next-gen sequencing, it's actually, you have to queue up in order to make it cost effective. And now you sit with that situation and you have multiple labs. Are you going to have sequences in every lab, which you have to maintain, which adds to massive amount of cost? How are you going to address that? Sorry, it's a bit of a curveball.
The ideal would be to get the sample preparation for sequencing as cost effective as possible, so that you can potentially, and as sensitive as possible, so potentially you could run it on something like a Flongle or a kind of a MySeq, well, not iSeq, iSeq or something, a small, low throughput sequencing device that can reasonably cost effectively kind of produce adequate data to make that early call. Like I think, I mean, at this stage, it's not there yet. I mean, you maybe, if you get the probe capture and the CRISPR both working well enough that you could use it on a mini flow cell, potentially you could do a run a day, maybe in a normal lab. But I mean, it again then comes to the matter that it's a little bit analogous to the GeneXpert, in the sense of how that all evolved, where originally, I mean, GeneXpert was not used as a routine for everyone. A large reason of why that occurred was because of a study that showed it was better. And even though it was more expensive than doing a microscopy and culture, because it was better, you can't then justifiably not do it. And then you essentially will say, well, okay, yes, it is costly, but how do we get it cost effective enough that we can actually run it routinely? And I mean, just in the news lately or recently, there's a deal between Cepheid and Nanopore. So I think I can see where this is going. So.
Yeah, just 1 second. Ruan Marais? Yeah. Yes. Rob Warren's doing a big trial of 3,000 TB on sequencing. Why you are not clever and asking him the same question? And let's see what he answer. Yeah. How would you do? How would you solve that problem, Rob? No. It's because today, as a very popular, we have 220 people online. So I just trying to be polite today.
I'm sorry about that. In our study, thanks for introducing it, Julia, we're doing both surveillance and real-time sequencing. So we try to batch a few surveillance with a real-time sample, and that makes it cost-effective.
So yeah, I mean, I think with TB, you know, you can basically think of clinical microbiology as, you know, time to results for phenotypic microbiology. It's like virology is over here, where it's like CMV will take like 42 days. Then TB is like here, kind of around about 10 days-ish for a positive culture. And then, you know, Enterobacterales is like 48 hours, 24 hours, 48 hours. Now, virology is lost. It's already fallen to molecular. There's very little culture that is on a routinely going there. TB is next. I mean, GeneXpert has already kind of taken over most of the testing, and now with whole genome sequencing, you can kind of get the remaining phenotypic culture kind of into you know, the phenotypic culture out of routine. And then I think once you get that 24-48 hour, you know, barrier, then the entire lab potentially could be funneled into a single system, and then I guess you get economy of scale very quickly.
Yeah, sorry, it's very traditional of our seminars that we grill the speaker after. So.
I see.
Thank you so much for your talk. I found it very interesting, and the amount of work you put in is clearly visible. I have more of a technical question rather than biological. And it relates to the API. So you send something you post, right? And then you get a request back. So in terms of security for the patient data, what measures are there to kind of stop me, if I were a hacker, to kind of just get that ID and kind of steal the data that comes back?
Yeah, so a lot of that comes from how I firstly see it deployed. So the tool that's to be used for clinical diagnostics, the goal is to deploy it in a like a fenced system, basically not connected to the internet, where you keep all your data locally. Obviously, there's permission systems and so on, so that within your facility, you're not having like inappropriate access. The deploying it on the cloud, at least initially, I see this more as kind of a research tool to expand access to kind of the algorithms and way of doing it. But I mean, I wouldn't initially deploy it as a web tool for clinical use. That's not the goal there. And certainly, I mean, doing that is possible, but that would be a larger team effort rather than just myself.
Thank you so much.
I have a question regarding the treatment recommendation algorithm. First, if this is already published, if this is published, is it open source? And if this is expandable for TB, for example, and other type of drug resistance for the recommendation, the final recommendation?
So it's not published yet, but probably, I mean, I'm writing it, so I'm hoping to submit it, I guess, sometime in the next 6 months. I plan to publish it, release the code with an MIT license. So you should be able to use it. And it is, I haven't used it for TB, but it should be usable for TB.
And the clinical interpretation guidelines, that for national guidelines, or you are mining that from the literature?
So it depends. Like if there are guidelines, then guidelines, it would default to the local guideline, basically. But in some cases there aren't, and so you would see there, for example, those Staph aureus is, you know, at the time of that version, I mean, that was the best reference or most appropriate reference to put in for that evaluation.
Okay, so just hold your question a little bit in person here. Yeah, let's go a little bit online. As I mentioned, we still have 215 people online. Yeah, so let's just here in the order. I will ask Melchiori, Betty. Yeah, I will unmute your microphone. Or to be honest, MB, I think that you have to unmute the microphone. So can you're just going to be unmuted now, and then you can ask your question, please.
Oh, okay, thank you. Yeah, my question is more technical. Yeah, first of all,
yes, you can go on.
Okay, thank you. Thank you for the good presentation and the innovation.
We can hear you. Try to talk again.
Okay, can you hear me?
Yeah, loud and clear.
Okay, thank you so much. Thank you also for the good presentation, but my question is more technical. It's all about the API, and yeah, you have explained it's pulling some information from the papers, from the previous papers, so that the clinician can understand more about the results. So I was trying to ask, how do you do that? How do you fetch those information from the internet using the API, so that the clinician can understand more of? And also, what about the requirements for the deploying that software in terms of capacity, computational capacity?
Yeah, thank you. And for our curiosity, yeah, where are you attending the seminar from?
I'm attending from Tanzania, from Kilimanjaro Clinical Research Institute.
Okay, thank you.
Okay, so I'll answer the second question first. So you could best consider the software similar to, I guess, a Galaxy. You can deploy it on any system, but because it is going to create containers in which to run specific tools, ultimately what resources the system is going to need is going to depend on what you want to run on it. So, you know, you could run it on a laptop with 8 GB of RAM, but then you would be limited to tools that need 8 GB of RAM, which, if that makes sense. So the tool itself is not the limiting factor. It's what you want to do with it that would be the limiting factor. You can choose whether it runs things in Docker or Singularity, so that you could also deploy it on an institutional HPC, where obviously the desire will be to use Singularity rather than Docker. And then in terms of the literature based, that is currently from a curated database. That is not the inference is not pulled in directly from the internet, right? So the more in-depth advice, on terms of duration and that type of thing, that has to, at this stage, is through a curated database. But in terms of choosing which drugs, those are automated algorithms and will work for any organism. So for every organism, it might not tell you you should treat for 7 days, but if this is one of the, for example, 100 or so relevant clinical pathogens, in most cases, that will be the case. I mean, yeah, I'm certainly thinking of ways in which to automate that process as well, but not there yet.
Okay, thank you. Yeah, one of the very impressive thing is that Ruan Marais is a clinician. Yeah, with programming. So Vin de Villers, yeah, you still have time. You are in the School of Data Science. We want to see your API programming with language.
That one is Python and the front end is in React.
In Python and React in 6 months. It's just a small joke.
No. I mean, it wasn't just 6 months. But the kind of inference algorithm, at this stage, I haven't really reached a speed barrier, but I think, I mean, if I do reach that, it'll need to be put into Rust. Yeah.
Okay, thank you. So the next question, Ambi, if you can unmute Musoka Abubakar, yeah, please. You're gonna be unmuted, and then you can ask your question. Yeah, Ambi. Yeah, sorry, Musoka. Yeah, you can unmute yourself. Yeah, now you have permission. Yeah. Okay, until he can unmute himself, can you give permission to Obid Etaba, Ambi? And then Ogbi, if you can unmute yourself, yeah, and ask your question. Yeah, thank you. Okay, and then we have Bismarck. Yeah, okay, go on. Yeah, kind of. Go again. Yeah.
This is Musola Abubakar from Kampala International University, Uganda.
Yeah, Musoka, we're having problem to hear you. Can you ask your question?
Can you hear me?
I think that, yeah. So, yeah, sorry, we couldn't hear you properly. Would you mind typing your question? It was very, it looked that you are in the top of Kilimanjaro. Yeah. Yeah, okay. So we have a very good question here from Samuel. You know Samuel? Yeah, he's in Indiana. Yeah. So I love the analogy to Gene Expert when dubbed too expensive, but then simply become necessary. I look forward to be part of diagnostic network optimization incorporating next generation sequence to find the sweet spot for essential use of NGS and coastal control. I don't know if you have some comment to Sam. Sam, it is a good collaborators that also work with us, yeah, from the US. Yeah.
No. Hi, Sam. Thanks for joining.
Okay. And then my last question would be, yeah. So let's just hear, yeah. And then it is from Arnold Abukara, that such innovative work, thank you. The mere presence of resistance gen may not necessarily translate to phenotypic drug resistance. How does this algorithm account for such case?
So it basically in classifying a detection base, it will either classify it as penetrant or not, where you have, when you are asking for your interpretation of your data, you can set your risk threshold basically. So I guess the example would be something like a AMP-C with a Sriracha species, Serratia marcescens, for example. So the risk of an AMP-C being induced is reasonably low with that, but you know, in some cases, you know, let's say you're treating a endocarditis, you know, you might consider that risk not worth taking. But if it's a urinary tract infection and the patient is already improving, you know, then you're likely to then it might be reasonable to continue the drug and not consider the induction. So in the interpretation aspect, you can set that kind of that level of risk that you are willing to accept when you are faced with a like gene that can be induced but is not necessarily. So I mean, you can't necessarily in all cases give what therapy you should do, but you know, you can basically say that, you know, at this risk level, this would be the appropriate thing, and at that risk level, that might be the appropriate thing. And in some cases, you might have genes that there is no information about, in which case they would be reported as present. And well, depending on the organism, depending on the site, you know, all of those factors are considered. So, I mean, it's a complicated question, but basically all information that can be integrated and interpreted is, but in some cases, there is just not the knowledge base to make a call automatically.
Okay, so just to thank for Ruan Marais, we also gonna come to 1 p.m. and we like to run this seminar like a Switzerland. It start midday exactly, finish at midday. Yeah, and we also want him to come back. Otherwise, he's gonna say they grill me so much that I will never come back there. So thank you very much. And before we finish, I have one important announcement. Lenin, can you stand and come here? Yeah. So it is a great honor to announce today that we have a new professor in the room. Yeah. So it was quite a long professor. She's already associate professor at University of KwaZulu-Natal, but today she's also officiated as a professor at Stellenbosch University at the Department of Biology. Thank you.
I need to make a speech.
No, just say whatever you want.
Thank you very much, Marais. I guess this was a wonderful presentation. Looking forward to your PhD and Professor after that. In addition